Welcome to our lecture on cell and molecular biology. Today, we are going to explore one of the most fascinating and fundamental processes in all of life, DNA replication. We will discover how simple bacteria copy their circular genomes with incredible speed, and contrast that with the massive logistical challenge our own eukaryotic cells face when copying long, linear chromosomes. By the end of this session, you will understand not just the mechanics of these processes, but also how they relate to bacterial infections, the aging of our bodies, and even cancer. Let us dive in. Now that we know our goals, let us start by looking at how simple bacteria handle this task. Imagine you have a perfectly circular rubber band, and you want to trace its inner edge with two pens moving in opposite directions. They will eventually meet on the exact opposite side. This is exactly how bacteria and archaea copy their genetic material. Because their DNA is a closed loop, there are no loose ends to worry about. The process starts at a single specific location, known as the origin of replication, or ORIC. From there, the DNA unwinds, forming a replication bubble. Two replication forks travel bidirectionally around the circle until they meet at the termination region. This elegant simplicity allows single-celled organisms to reproduce at astonishing speeds without losing any genetic information. Understanding this circular replication is not just an academic exercise. It has profound real-world applications. Why does this matter to us? Because when a patient suffers from a severe bacterial infection, those bacteria are multiplying by rapidly copying their circular DNA using the exact method we just discussed. Many of our most effective antibiotics, such as fluoroquinolones, are designed to specifically target and jam the molecular machinery that separates these interlinked circular DNA copies, effectively stopping the infection in its tracks. Furthermore, in biotechnology, scientists harness small circular DNA molecules called plasmids to clone genes, allowing us to mass-produce life-saving medicines like synthetic human insulin. Now that we understand the biological mechanism, let us look at how we can mathematically model the speed of this process. The formula shown here calculates the total time required to completely replicate a circular chromosome. In this equation, T represents the total time. C stands for the circumference, which is the total size of the genome measured in base pairs. R represents the rate of DNA synthesis at a single replication fork, measured in base pairs per second. Notice that we divide by 2R. This is a crucial detail because replication is bidirectional. Two forks are moving simultaneously, effectively doubling the overall copying speed. If the genome size C increases, the time T will naturally increase. Conversely, a faster polymerase rate, R, will decrease the total time required. Before we move on to more complex organisms, let us see this formula in action with a concrete example. We're looking at the bacterium E. coli, which has a genome size of 4.6 million base pairs. Its DNA polymerase operates at a remarkable rate of 1,000 base pairs per second. We are asked to find the total replication time. We choose our circular replication formula because E. coli has a single circular chromosome. We substitute the genome size for C and the rate for R. The denominator becomes 2,000, representing the combined speed of the two forks moving away from the origin. Dividing the total base pairs by this combined rate gives us 2,300 seconds. Dividing by 60 gives a final answer of approximately 38.3 minutes. This physically means that under optimal conditions, E. coli can completely copy its ENA in under 40 minutes, explaining why bacterial populations grow so incredibly fast. Having calculated the speed of this process, Let us visualize exactly what is happening at the molecular level. Here, you are looking at a classic theta structure, which represents circular DNA mid-replication. The large blue circle is the original bacterial chromosome. At the top, you can see the replication bubble that is opened up at the origin of replication, labeled ORI-C. Inside this bubble, the red lines represent the newly synthesized DNA strands. Notice the two black arrows pointing in opposite directions, away from the origin. These represent the two replication forks moving bidirectionally. As time passes, these forks will continue to travel around the entire blue circle until they meet at the bottom, resulting in two complete, separate circular genomes. Now that we have mastered the bacterial strategy, we must address the eukaryotic challenge. Imagine you are tasked with copying a massive encyclopedia by hand in just a few hours. If you start at page 1, you will never finish. Instead, you would hire hundreds of assistants to work simultaneously. This is exactly the strategy eukaryotic cells, like our own human cells, use to copy their linear DNA. Because our chromosomes are incredibly long, straight strands, starting from just one end is impossible. Instead, eukaryotic chromosomes contain thousands of distinct origins of replication scattered along their length. At each origin, a replication bubble forms, and forks move outward bidirectionally. Eventually, these adjacent bubbles meet and fuse together. This multi-origin parallel processing is absolutely essential to replicate massive genomes within the short time frame of a cell cycle. To truly appreciate this multi-origin strategy, we can mathematically model the replication time for a linear chromosome. This formula is crucial for understanding how complex cells manage their strict biological time limits. Here, T is the total time required, L represents the total length of the linear chromosome in base pairs, N is the number of active origins of replication, and R is the rate of the DNA polymerase. Just like before, the factor of 2 accounts for the bidirectional nature of each replication bubble. Conceptually, this equation shows us that if the number of origins, N, increases, the total time T decreases dramatically. This is exactly how eukaryotes compensate for having a much slower polymerase rate compared to bacteria. They make up for slow speed with massive parallel processing. Let us apply this formula to see exactly why a single origin would be catastrophic for complex life. We are looking at human chromosome 1, which contains approximately 250 million base pairs. The eukaryotic DNA polymerase operates at a relatively slow rate of 50 base pairs per second. We are asked to find how long it would take to replicate if it only had a single origin. We use our linear replication formula and intentionally set n to 1. We substitute the length for L and the rate for R. The denominator becomes 100, representing the combined speed of just 2 forks moving away from one hypothetical origin. Dividing the massive length by 100 gives us a final answer of 2.5 million seconds. Physically, this represents an enormous mathematical bottleneck. Following up on that massive number of seconds, let us convert this result into a time frame we can intuitively understand, proving our biological concept. We take our result of 2.5 million seconds and divide it by 86,400, which is the number of seconds in a single day. The calculation yields approximately 29 days. This mathematical step physically proves that a human cell cannot possibly rely on a single origin. It would take an entire month just to copy one single chromosome. In reality, a human cell completes this entire process in about 8 hours. The only mathematical and biological way to bridge the gap between 29 days and 8 hours is by utilizing thousands of origins simultaneously. This perfectly illustrates the absolute necessity of parallel processing in eukaryotic life. Now that we have proven the mathematical necessity of multiple origins, let us look at how this appears structurally. In this diagram, you see a long, straight blue line, representing a segment of a linear eukaryotic chromosome. Instead of just one bubble, you can clearly see 3 distinct replication bubbles forming simultaneously along the DNA strand. These are the multiple origins of replication we just calculated the need for. Inside each bubble, the red lines show the new DNA being synthesized. Notice the black arrows at the edges of every bubble? They show bidirectional growth. As these bubbles expand outward, they will eventually crash into each other and fuse. This simultaneous firing of multiple origins is the only way complex organisms can successfully divide and grow in a reasonable time frame. While multiple origins solve the speed issue, being linear introduces a new severe complication known as the end replication problem. Imagine you are painting the floor of a room and you slowly back yourself into a corner. When you reach the very edge, you cannot paint the exact spot you are standing on without stepping outside. Linear DNA faces a very similar dilemma. DNA polymerases cannot start copying from nothing. They require a pre-existing starting block, usually a short RNA primer. However, when the final primer at the very end of the linear chromosome is removed, there is no upstream DNA left for the polymerase to grab onto and fill the gap. Because of this physical limitation on the lagging strand, the very last tiny segment of DNA cannot be copied. This means that every single time a cell divides, its chromosomes get slightly shorter. Since we know DNA is lost during every division, how do our cells survive this constant erosion? The answer lies in telomeres. To protect our essential, life-giving genes from being destroyed, the ends of eukaryotic chromosomes are capped with these telomeres, which are long, repetitive, non-coding DNA sequences. They act as disposable buffers. However, this means telomere shortening acts as a biological countdown timer. This gradual loss is one of the primary molecular causes of aging. When telomeres become critically short, the cell hits what is known as the Hayflick limit and permanently stops dividing to prevent genetic damage. Conversely, this mechanism is deeply tied to cancer. Cancer cells often hijack a special enzyme called telomerase, which artificially rebuilds these lost ends. This grants cancer cells biological immortality, making telomerase a major target for modern cancer research. To understand this biological clock ticking inside our cells, we can use a linear decay model to calculate telomere shortening. This mathematical model helps researchers predict exactly when a specific cell lineage will enter senescence. In this equation, L subscript n represents the final length of the telomere after a certain number of divisions. L subscript 0 is the initial length of the telomere before any divisions occur. The variable n represents the number of cell divisions that have taken place. Finally, delta L represents the specific length of DNA lost during a single replication cycle. Conceptually, as the number of divisions, n, increases, the subtraction term grows larger. This causes the final telomere length to steadily decrease over time, perfectly modeling the molecular aging process. Let us apply this decay model to calculate the actual lifespan of a human somatic cell, known as the Hayflick limit. We are given a cell that starts with an initial telomere length of 10,000 base pairs. During each cell division, the end replication problem causes a loss of 100 base pairs. The cell will permanently stop dividing when its telomeres reach a critical length of 4,000 base pairs. We are asked to calculate how many times this cell can divide. We choose our telomere shortening formula and identify our variables. L0 is 10,000, delta L is 100, and Ln is 4,000. We substitute these values into the equation, setting up a simple linear equation where the only unknown variable is n, the number of divisions. This establishes the exact endpoint of the cell's life. Now that we have our equation set up, let us walk through the algebraic reasoning to derive the cell's exact lifespan. We start with our substituted equation. Our goal is to isolate n. First, we rearrange the equation by subtracting 4,000 from 10,000. This subtraction is physically meaningful. It tells us that the cell has a safe buffer of exactly 6,000 base pairs before it hits the danger zone. We set this buffer equal to 100n, which represents the total loss over time. Finally, we divide the 6,000 base pair buffer by the 100 base pairs lost per division. This gives us a final value of 60. We have mathematically proven that this normal human cell can undergo exactly 60 divisions before its telomeres become critically short, triggering senescence. This is the quantitative proof of the Hayflick limit. As we conclude our lecture, let us review the most important ideas we have uncovered today. First, we saw that bacterial circular DNA replicates highly efficiently from a single origin, allowing for rapid population growth without any loss of genetic material. Second, we learned that eukaryotic linear DNA must utilize thousands of origins simultaneously. Without this parallel processing, complex life simply could not exist. Finally, we discovered the cost of being linear, the end replication problem. Because polymerases cannot copy the very ends of DNA, our chromosomes shorten every time our cells divide. Telomeres absorb this damage, but their eventual depletion acts as a biological clock, driving the aging process. You now have a comprehensive understanding of both circular and linear DNA replication. Excellent work today.